Okay, are we ready?
Yes.
Okay.
So I'm ReidAnn.
I got to do my intro.
Sorry.
You're stepping on my intro.
I'm sorry.
This is Mr. Anico. We are back with another interview for Scaluda 21. This is with ReidAnn Seaver, and she's here to talk with us about getting your first job as a researcher. And ReidAnn, why don't you give us that like a minute synopsis to talk us through high school to your current position?
Yeah, so I graduated Norrin in 2017, and I attended Hood College in Frederick, Maryland for 4 years. I was a major in biology and French, and I minored in chemistry. Had a couple of internships over the course of my time at school, as well as a senior thesis on how climate change and water temperatures are affecting American lobster populations, and was fortunate to start my job as an intern this past summer through an intern placement program that was supposed to happen during COVID, but was unfortunately rescheduled. And then 2 weeks into being an intern, my boss saw something important in me and asked me to stay full time. So here I am.
And then, so actually your internship getting delayed maybe wasn't a bad thing because it pushed you late enough that you'd be actually in a position to accept that job, sort of. So.
Yes.
Silver lining. So what kind of firm are you working? Is it a private company you're working for now, or?
No, I work for the University of Pittsburgh, but we are a part of a lab that is at Children's Hospital. So I technically am an employee of the university, but all of our collaborations are specifically within pediatrics.
Okay. You have plans to go back to school for medical school, right? And
yes.
You want to real quickly, like in 30 seconds, summarize the philosophy behind a gap year, because it's actually going to show up in at least one other video, actually.
Yeah, so a lot of students previously were taking gap years before med school just as a chance to reset and work experience, to not be a student for 9 years straight. But in my case, it's becoming a more popular thing because they have now changed the first 2 years of med school to be pass-fail. So a lot of residency programs after you graduate med school are looking at your research experience as a way to place you into programs. So I had planned on taking 1 year to do that kind of relax and work a little bit, and then ended up deciding to take a second to make sure that I have the experience I need to get a good residency.
It's important to note, again, the work you're doing during your gap year is relevant and like intellectually rigorous, and it's you're not like laying on the beach or anything like that. It's extensive. So
it's a full-time research position. So.
That's exciting. And that's what we're going to spend the rest of the time then talking about. So is the route that you did to get this like typical? Like, is it usually you have to have some sort of foot in the door, like through an internship, or do you just like apply for research jobs like you would on like monster.com or something?
There's a little bit of both. So for myself specifically, the lab that I work in is kind of prestigious topic area because it is children's brain cancer. So having that foot in the door, being an intern first, is definitely the way in. But research assistantship and technician positions go up all the time on specifically Pitt's website, but as well for other labs in the university. So in my case, to get into a really technical clinical lab, you do kind of have to have some internship experience, not necessarily with the same lab, but at least prior to your first job. But there are other jobs that, their only requirements are a bachelor's of science or a bachelor's of arts within a science field, and you apply the same as anybody else.
You're a new researcher. You're fresh out of college. This is your first full-time professional job. What's a typical day like for you as a researcher?
The best way I can describe a typical day is that no 2 days look the same, and that's the best part of the job. Is you have small routine things where you're sometimes in and out at the same time every day, and you always have little things like lab maintenance, but it really depends on the project you're looking on. So for myself right now, I have a lot of cell and mouse work going on. So I'll come in by 8:49 in the morning. I spend the first probably hour of my day just doing inventory of the lab, catching up with my coworkers, having kind of, my boss likes to do morning chats with everyone for 5 minutes just to make sure they need everything or have everything for the day. And then I typically go down and check my mice first thing in the morning, say hello to them, make sure they're doing good. And then I come back up, and the next couple of hours up until lunch usually are a mix of data analysis at my desk or helping out a coworker with their project. And then I usually spend my afternoons doing cell culture work. And you can leave as early as 3:30 if you have things done, or sometimes you're there till 9 p.m. if you have flow cytometry. It really varies. But especially in research, because your schedules are so different, the time is very forgiving. So my boss is an advocate of, if you have to stay 4 hours late, the next day you leave 4 hours early, and trying to make, even though research isn't built around a 9 to 5, you can still maintain other fun life activities outside of the job.
I mean, I'm sure we've all had to stay late for flow cytometry. I mean, it's like,
yeah, it's an everyday thing.
So there's so many good things to touch up on that. All right, so let's see. So in terms of, so you mentioned the idea that, again, the research experience, a lot of times you're at the will of the experiment. If you're running some sort of data analysis program, or you're conducting some sort of experiment, you're kind of there until the experiment's done.
Yes.
So, but in the same regards, sort of the academic lifestyle is kind of forgiving that way, in terms of, everybody's really focused on the results, and not so much that everybody necessarily needs to clock in at the exact same time. So you've mentioned mice a couple times. So let's bring that up real quick then. So the work that you do uses like animal test subjects, basically? Is that the idea?
Yes. Yes, we do. So since we are a cancer lab, we have to use a primate model before anything we do goes to clinical study, especially because it's with kids. So the anatomy of the mouse brain is the most similar to a child's. So we use mice, both for brain work and for whole body work. So I actually have a picture. If I can.
You're gonna have a mouse.
I know, I don't have a mouse with me. No, I'm not done the animal silly, but if I can share my screen, I think you have to give me the pattern.
Oh, there it is. All right, let's see here.
So these 3 little pictures. So this little mouse here in the middle is the type that I work with directly. So these are hairless and immune systemless mice. And
whoa. Let's review that again. So is this a breed of mice?
Yes, it's a specific strain that was created in another lab at the cancer center, and then we grow and raise these mice.
So they have no hair, and they have
no hair,
no functional immune system.
No, well, they have a very slight functional immune system to prevent things like a common cold from getting them sick, but no functional immune system.
Wow.
And because of that, they don't have a strong fight or flight instinct. So very easy to handle in the lab. They don't bite. As you can see, this one's just sitting on my hand, and she was there for probably 5 minutes as I was working with her, which is one of the benefits to be able to use this mice. The picture to the left of the mouse is actually a picture of the cells line that I work with. So they're called neuroblastoma, which is a tumor that is neural tissue, but actually grows on the adrenal gland on top of the kidney. So this is a little screen grab of one of our microscopes with these cells. And then the picture on your right is actually what it looks like when we grow them. So our lab doesn't use the stereotypical, petri dish with agar that smells really bad, and you leave it in a shaker plate overnight. So this is actually a 37-degree CO2 incubator. And so all of our cells grow in these little plastic flasks with liquid media inside of them, because it more similarly represents the tumor environment within the animal or a person. And then we'll do things with these where we take cells out of a mouse that has a tumor to try to figure out different treatments, or sometimes we actually give the mice tumors via injection to look at things like survival studies, surgery treatments. Can we boost their immune system with peptide vaccines to have them learn to fight the disease on their own? So there's all different kinds of experiments going on, but mine in particular with those neuroblastoma cells and then these immune systemless mice.
Okay. That's a lot.
It is a lot.
There's a lot of things you guys are doing there. Oh, thanks for sharing that. That's awesome. All right. So in terms of the research itself, I would imagine a lot of kids watching right now are probably asking, is it tough to be a scientist working with like live animal test subjects? Is that like, is it hard to do, like, just because they're cute and kind of
they're very cute, and it's definitely not for everybody. One of the first things my boss told me was, you cannot name them because you will get attached. But in absolutely no lab on the face of this earth are you ever obligated to work with animals. So if you know that you're one of those people that they're just too dang cute and you can't do it, you can always opt out and do other types of work. The first day you work with the mice is very strange because you're always worried about hurting them. But once you get used to the idea, they're very simple, and it's just a lot of making sure that you're being gentle enough with them and treating the mice correctly. And there are lots of protocols in place as well that we have to follow from the university, the USDA, NCI, the National Institute of Health, and all these other places to make sure that even though we're doing experiments on mice, the mice are given the best housing and food and life situations that they can.
All right, thanks for sharing that. That's great. So looking back, like what has surprised you the most about being a full-time salaried researcher that you wouldn't have thought, a year ago?
Yeah. So when I came into it, I had the expectation that I'm sure most people do, where it's, you're the fresh college graduate, you are the bottom of the food chain, you have a bachelor's, you don't have a master's, you don't have a PhD, and you're not actively working for either of those. So, my expectation was I'm going to work for someone else's project and hopefully put my name on something by the time I leave. And that has not at all been the case. And from what I've seen with other labs is almost never the case as well. So I came in my first 2 weeks, learn how to do this, make sure you know how to use that. Here's where this is. And as soon as that time point went over, my boss was like, here's your project, time to get started. So, even though I don't have a PhD and I don't have a master's, there are people within each lab who have a very particular type of research or a cell line or a machine that they're just really good at using. And that becomes your thing. So we have another post-bac in my lab who is the single cell RNA sequencing guy. He doesn't have a PhD. He doesn't want to get a PhD, but people from other labs and other states ask him to do this work for him. So you think that coming in as a new graduate with not a lot of experience, that you're just going to kind of be fed bits and scraps, but you get a pinhole, that's your thing, and you become in charge of something, and it really helps you to grow as a researcher.
Can you talk about how, and again, in terms of what you're allowed to share, like how is what you're learning from mice going to be applicable to kids?
Yeah. So the way that the field of cancer research is now shifting is away from chemo, because we know chemotherapy is very harsh in the body and has a long-term effect. The work that we're doing in mice is try to use immunotherapy and peptide vaccines instead of having to treat with harsh drugs after the cancer has already been diagnosed. So we are able to try out these systems in mice without immune systems, which mimics how a kid is when they're sick because their immune system is knocked down by their cancer. And we're able to try out these new therapies that don't involve harsh drugs or having to be on chemo for 2 years or having to live in a hospital, things that can essentially, instead of treating the body that already has cancer, teaching the body that's been diagnosed to fight it on its own. And that's where it's going. And that's what a lot of these mice projects are about now is, can we find new ways that once a tumor pops up, we can actually teach that body's immune system to fight it on its own instead of having to pump it full of drugs and hoping for the best.
So, sounds like rewarding in and of itself. And again, a great stepping stone into medical school. Mm-hmm. In a couple of years with that background as well. If you could, talk to ReidAnn back, 8 years ago or whatever like that, is there like one piece of advice that you would offer yourself, just in general, between there and here, to sort of help smooth things along the way?
Yeah, I would say just because you're not great at the science subject now doesn't mean that you can't grow into it later. AP Bio, I was not Mrs. Anselmino's favorite by any means. I graduated top of my class with honor cords and degrees and job opportunities. So just because you're not good at it now or you don't understand it the first time you see it does not mean that it can't become your passion.
That's excellent. Well, again, it's exciting work that you're doing, and a stepping stone to, I'm sure, even more exciting things in the future. Hopefully the kids realize that science is an amazing stepping stone to lots of crazy opportunities. So
yeah,
it's excellent. Hey, well, again, thanks for your time. Hope you have a good holiday break, and we appreciate you sharing your experiences with us.
Of course, thank you.